Welcome to the podcast of Hemisphere, the official journal of the European Hematology Association. Hemisphere's podcast presents insightful, expert discussions about recent hematology publications. We hope you enjoy.
Hello and welcome to the Hemisphere podcast. My name is Charles de Bock and I'm your host. Myelodysplastic neoplasms are blood disorders that rob patients of healthy red blood cells and for many, inching towards acute myeloid leukemia. Allogeneic hematopoietic cell transplantation, or allo-HCT, is curative but is often reserved for high-risk MDS patients and not low-risk MDS patients, which accounts for almost 2/3 of all patients. Instead, these low-risk MDS patients receive supportive care like red cell blood transfusions or erythropoiesis-stimulating agents, but these are only offering temporary relief, and so there's a real need to find new therapeutic options for these patients. And on this note, it is with great pleasure that I introduce two researchers from the University of Dresden in Germany, and this is Professor Manja Wobus, who is head of the Stem Cell Research Lab, and Dr. Katja Sockel, who is the senior physician and head of the MDS MPN program, and they are the first and senior authors on a brand new publication in Hemisphere with the title Preclinical Efficacy of tasquinimod in Myelodysplastic Neoplasms, Restoring Erythropoiesis and Mitigating Bone Loss. I welcome you, and I've realized I probably said that drug completely wrong, if it's tasquinimod or is it just task? Maybe you can correct me. How do we say that drug? Is it tasquinimod? Tasquinimod. Okay, so we've got that one up. Or I'm going to call it task for short, maybe is easier. Welcome to you both. Actually, we just published our most recent podcast where we actually focused on myeloproliferative neoplasms. And for this podcast, we are focusing on myelodysplastic syndromes. And both are really bone marrow disorders. But whereas MPNs have an overproduction of mature blood cells, MDS are the opposite. And there's ineffective production of blood cells where patients suffer from severe anemia and often have infections. So I mentioned that red blood cell transfusions are the current standard of care in low-risk patients, but perhaps you can provide some additional insight into the challenges of treating these patients.
Yes, so are many challenges in MDS. So first of all, there are many patients out there living with MDS, as it is one of the most common hematologic neoplasms in the elderly. And even so, we classify some patients as lower risk. They often experience a substantial symptom burden, such as transfusion-dependent anemia, fatigue, as you have already mentioned, infections, bleeding complications, which strongly affect the daily life of our patients. So therefore, in clinical reality, this is often far away from being a truly low-risk disease. So fortunately, over the last years, we have made significant progress in understanding the disease, and we now have far more therapeutic options available than we did 5 years ago. So we now have drugs like ESA, we have lenalidomide for a long time, but only for patients with deletion 5q. We have luspatercept, an erythrocytic methylating agent available, and more recently we have imetelstat, but only for selected transfusion-dependent patients. However, most of these treatments are primarily symptomatic, so they can be very effective in improving cytopenia and also in reducing transfusion burden, but they generally do not modify the underlying disease biology. And of course, only a subset of our patients will respond to these treatments. And even among the responders, many patients will lose their response over time. So therefore, what we really need are therapies with more durable effects, therapies that not only address the consequences of ineffective hematopoiesis, but actually modify the disease itself and potentially prevent progression to high-risk MDS or even AML.
Okay. So it's interesting in your paper, like talking about disease biology and understanding it more. In your study, you focus on a protein called S100A9, which is a danger-associated molecular pattern or DAMP molecule. Can you probably, for our readers and listeners, introduce what S100A9 is and what led you to focus on this particular molecule in the context of MDS?
Yes, absolutely. So it is indisputable that dysregulated inflammatory processes within the bone marrow microenvironment, the so-called bone marrow niche, play an important role in the impaired hematopoiesis and disease progression in MDS. So S100A9 is particularly interesting in this context because it appears to be one of the key drivers of this pathological inflammation. So it belongs to the S1 family of inflammatory proteins and is mainly produced by myeloid cells, such as monocytes and neutrophils. So functionally, it acts as a so-called DAMP or damage-associated molecular pattern. These are endogenous danger signals that activate the innate immune system and promotes inflammatory responses during, let's say, cellular stress or tissue damage. So several groups have shown that inflammatory signaling pathways are chronically overactivated in MDS, especially in the early low-risk MDS patients. And this pro-inflammatory environment gives the malignant clone a selective advantage and promotes genomic instability, thereby contributing to disease progression on the one hand. And at the same time, the inflammatory cytokines suppress also the normal, the wild type hematopoiesis by inducing pyroptotic cell death, which lead to the cytopenia and the bone marrow failure in our low-risk MDS patients. By binding to receptors such as toll-like receptor 4, S100A9 activates the downstream inflammatory signaling pathways involving IRF4 and TRAF6 and NF-kappa B. And this ultimately lead to the inflammasome activation and further production of pro-inflammatory cytokines within the bone marrow microenvironment. So therefore, targeting this S100A9-driven inflammation very early in the inflammation cascade seemed to be a very promising approach to us.
Okay. That's fantastic, because I think from an outsider's perspective, I would have assumed the expectation was to be looking at and focus on the defective stem cells per se and not the inflammation in general, because you'd be going, that might be an easier target. But here you investigate in your paper the role of the mesenchymal stromal cells in the context of the bone marrow inflammation. So how does your paper change the way we think about MDS and its treatment?
You're right. Traditionally, MDS has been viewed as a disease of the hematopoietic stem cell compartment. But since several years, there's a significant contribution of the bone marrow microenvironment to disease progression and also to treatment response are demonstrated by several investigations and several groups. Context, the mesenchymal stromal cells, or in short MSCs, play an important role as they support hematopoiesis and immunomodulation. The key conceptual shift is that MDS may be sustained not only by the intrinsic genetic defects in the malignant cells, but also by a dysfunctional inflammatory or microenvironment or niche that supports the disease persistence and the clonal evolution. The MSCs appear to participate in creating a self-reinforcing inflammatory loop within the bone marrow. And that means that even if we target the malignant clone efficiently, the diseased microenvironment may continue to promote the ineffective hematopoiesis and resistance. Our findings support the idea that MDS should be considered as an ecosystem disease rather than purely a cell-autonomous stem cell disorder. Therapeutically, this opens the possibility of combining clone-directed therapies with approaches that target the inflammatory microenvironment or the stromal dysfunction. And it may also help to explain why some patients do not fully respond to any therapy, or why the inflammation-associated symptoms persist despite cytoreduction. In the future, MSC-related inflammatory signatures could potentially serve as biomarkers for disease activity, for the progression risk, and also for treatment response. And more broadly, targeting the bone marrow microenvironment could allow us not only to suppress the malignant clone, but also to restore a healthier hematopoietic microenvironment and improve thereby the normal blood function.
I always find that quite intriguing because as humans, we like to use reductionist models and we hone in on the easiest target, which is, okay, there's an intrinsic defect in the HSC. That is what we need to fix, and forget about the niche and the ecosystem, as you quite likely nicely put. And whenever I hear this self-inflammatory loop, propelling, it's like that's one starts to recharge the other, and then you get into this vicious cycle. So yeah, hitting this at multiple levels, intrinsic and extrinsic, seems like a fantastic way to move forward. And in your study, of course, you do that. You start with not a new drug, but you repurpose a drug from a different, and we can talk about that. And I'm going to say it again, tasquinimod, or TASQ, TASQ for short, which is an immune modulatory agent, which was actually trialed in prostate cancers, as well as multiple myeloma and myelofibrosis. So what attracted you to this particular drug and its mechanism of action where you thought this might be perfect for MDS?
Yes. So what really attracted us to tasquinimod was the idea of targeting inflammation at a very central and very early point within the disease biology. So we started looking for compounds that could specifically interfere with S109-mediated inflammatory signaling. And that's how we came across tasquinimod, which is a small molecule drug with S109 inhibitory activity, which has demonstrated, as you have already mentioned, anti-angiogenic, anti-tumor, and immunomodulatory properties in a broad range of preclinical solid tumor models. And at that time, tasquinimod had already been investigated in prostate cancer clinical trials, even in phase 1, 2, and 3 trials. There was already substantial clinical experience with the compound, and it was well tolerated. So mechanistically, tasquinimod binds to S109 and inhibits the interaction with the toll-like receptor 4, thereby dampening downstream inflammatory signaling pathways, including NF-kappa B activation and inflammasome signaling. And in addition, tasquinimod appears to have broader immunomodulatory effects and has also been linked to targets such as HDAC4, which may further contribute to its biological activity in MDS. So, epigenetic dysregulation is also a key point in the MDS pathogenesis. And yes, another very practical advantage is that the drug is orally available, which is obviously highly relevant in our typical older MDS patients. And so we contacted the company who holds a patent for tasquinimod to ask whether they would be interested in supporting experimental work in MDS and providing the compound for us. And yes, after seeing this promising activity and let's say calming inflammation in our in vitro models, we became increasingly excited and decided to move forward with the mouse models. And you are fully right. Now the drug is also investigated in multiple myeloma and also in myelofibrosis.
Yeah. I do like the fact that you had to actually call the company as well and say, hey, I think we've got something exciting. And that they were willing to give the drug, because that's not always the case if it's off-label, which is fantastic. So one of the interesting findings in your paper is that TASQ actually decreases PD-L1. I mean, you look at a lot of pathways, I understand that, but that caught my eye because it decreased the levels of PD-L1 on MSC cells. And whenever I think PD-L1, I think immune cancer therapy, checkpoint inhibitors. And, you highlighted that, and I'm just wondering what your thoughts are of what that means in the context of MSCs and blood production for MDS patients, if anything.
So PD-L1 is typically discussed in the context of tumor evasion, but in the bone marrow niche, its role it's likely broader. and more immunoregulatory. And we had a previous study in which we observed that PD-L1 expression is involved in suppression of T-cell activity in the context of AML, which is really important also for treatment strategies like CAR T-cell therapy and such options. So in MSCs, the elevated PD-L1 may contribute to an immunosuppressive microenvironment that alters the normal hematopoietic support functions. As MSCs are key regulators of hematopoietic stem cell maintenance and immune homeostasis in the bone marrow, when they become inflammatory and express high levels of PD-L1, they might suppress the beneficial immune surveillance while simultaneously promote the dysfunctional hematopoiesis. And our finding that tasquinimod reduces the PDL expression on MSCs suggests an interruption of the inflammatory crosstalk between stromal cells, immune cells, and hematopoietic progenitors, which might support a reprogramming of the stromal compartment towards a less inflammatory and less suppressive state. And this could improve the functional support of the MSCs provide to normal hematopoiesis while reducing signals that favor the persistence of the malignant clone.
Yeah, that's amazing.
Our findings reinforce the idea that the immune checkpoint molecules in MDS are not only relevant on the malignant hematopoietic cells, but also within the stromal microenvironment itself.
I really like that. I like these unintended consequences of targeting one molecule but having a dual effect, as in making it not particularly comfortable for the malignant clone or the pre-malignant clone, but also helping support the wild-type HSCs to repopulate. That's really great. You mentioned you did the preclinical studies in vitro and then also in the mouse models, and you compared, in this particular case, you're looking, of course, for a therapeutic window. It's a nice drug. It's orally available, which is fantastic, but of course, you want to make sure it's safe for both healthy and diseased tissue. And you compared the administration of TASC in your transgenic mouse model, which was a NUP98 HOXD13 mouse model, which has MDS-like characteristics, and you compared the treatment between the wild-type lidamates and the MDS, or the NUP98 HOXD13. What was the most surprising finding that you had in these studies when you looked within the preclinical mouse model?
Well, in our mouse model, which it's in short termed NHD13 mouse model, we observed a significantly increased red blood cell count and hemoglobin levels after the treatment with tasquinimod, while no significant changes in the wild-type animals were observed. And that therapeutic window was particularly encouraging because one of the major concerns in MDS treatment is further damaging residual healthy hematopoiesis. And another interesting aspect was that the microenvironment itself appeared surprisingly kind of plastic. So even in a genetically driven MDS model, tasquinimod treatment resulted not only in the modulation of hematopoiesis, but also in an improved bone volume, which potentially includes an effect on MSCs as bone cell precursor cells. This supports our idea that the disease niche is not irreversibly fixed, but may remain therapeutically targetable. And moreover, our findings reinforce the concept that targeting the bone marrow microenvironment could complement clone-directed therapies without causing excessive toxicity to the normal marrow function. So together, our in vivo study also suggests that restoring the niche balance may be achievable without a global suppressing immune system therapy and impairing thereby healthy blood production.
And the doses that you gave the mouse or the in vivo, they're clinically achievable in humans? It wasn't like an outrageous dose? Did you do that comparison?
So we used published data on that, and this is comparable. Yes, it is.
Amazing. So I know you mentioned the bone volume, and I looked in your methods, and because you said you had the topic of bone strength, and I'm a bit of a nut for looking through the methods because I always find it interesting how people analyze mice. And you did this by isolating the vertebrae of the mice. And I think you essentially had some sort of jig where you squashed the in a mechanical press to measure the strength of the bones and the bone volume. I find that wild. And I just want to ask you a bit about that and how challenging it was. And was this a student that you gave, or you worked with a collaborator who had a jig that was made, or how did you think to do a squashing of a vertebrae to understand bone strength?
So all these experiments with the bone of the mice were performed by our colleagues from the Bone Lab, who are the experts on bone experiments here at the Dresden campus. And luckily, we have a long-term collaboration. It's very close to our institute and clinic. Yeah, it's a really fruitful collaboration because they are focused on the bone, and we are on the inside, on the bone marrow microenvironment. And so it complements each other. So the machine which was used for those experiments is a commercial one, and it's usually used for material testing. But in that context, it has been used to test the bone strength. And what was really technically demanding was the size of the murine vertebrae, because they are very small and fragile.
I can imagine they're very tiny.
Exactly. And you need a very consistent sample preparation, including the isolation, the cleaning, and the positioning in the machine to obtain reproducible measurements. And another important aspect was preserving the native biomechanical properties of the bone during preparation and storage, because dehydration or tissue damage can alter the compressive strength of the bone. So these experiments were really valuable because they provide a functional readout. If you apply only imaging techniques, you can show structural changes. But the mechanical compression test tells us whether those changes actually translate into an altered bone stability and strength. And these are important readouts also for the MDS patient treatment at the end.
Yeah, I find that really fascinating. And I mean, you do all that work and it becomes 2 lines in a paper. I must say that it's like the technical challenging part of that. It always seems simple, but thank you for explaining that, because I have dissected mice before and I know how small those vertebrae are, and to isolate them and do that, I can imagine, was very technically challenging. But as I've said before, we don't do things because they're easy. We do them because they're important and we need to know. And I agree. Imaging tells you one story, but combining that with a functional readout is very powerful. Okay, so the results are impressive, but they are in a mouse model. So this is a criticism that we all get when we do these models. I mean, all models are wrong, but some are useful. It's a famous quote. So my question really is, how closely does the NHD13 mouse model mirror what we see in the clinic for MDS patients? And what are some of the limitations that we should be thinking about when we read your study?
Yeah, that's a very important point, because no mouse model can fully recapitulate the biological and clinical heterogeneity of human MDS. So there are several MDS mouse models, including TET2, ASXL1, SF3B1, RANKS1, and spliceosome mutant models, and each captures different aspects of the disease. The NHD13 model is valuable because it reproduces several hallmark features of human disease, including ineffective hematopoiesis, cytopenias, and a slow progression towards acute myeloid leukemia. At the same time, MDS in patients is extremely heterogeneous, and not all patients show the same degree of bone marrow inflammation, inflammatory signaling, and it differs across the molecular subtypes and also between disease stages. Another important consideration is the mode of action of tasquinimod, besides S100A9 inhibition. It may also exert additional effects, including through HDAC4 related pathways or broader modulation of inflammatory signaling networks. That means we should probably think of tasquinimod not only as a single pathway inhibitor, but as a compound with potentially pleiotropic effects on the inflammatory microenvironment. I think that our study highlights both the strengths and the limitations of animal models. They allow mechanistic insights into which could possibly obtained in patients, but they also represent a simplified version of a highly heterogeneous human disease.
Yep. I mean, this is always the challenge. We find these findings, we use our model, but of course now we need to think about translation of tasquinimod into the clinic for low-risk patients. And so my question is, what are the next steps that you're planning? And you mentioned the heterogeneous disease and that not all MDS patients have high inflammation. And so would you classify or stratify patients based on high levels of S100A9? Is that a biomarker for its use in patients in the future? And what are you thinking about to move the dial forward in terms of translating tasquinimod for MDS patients, perhaps?
Yes. So, of course, our next major step is to bring this concept into the clinic. So we are currently preparing a phase 1/2 proof of concept trial of tasquinimod in patients with low risk MDS, which would also give us the option to evaluate the effects of tasquinimod in vivo in humans. So we've decided to focus on the early low risk MDS disease because this is where the pro-inflammatory bone marrow microenvironment seems to be the most relevant. And of course, the key question is whether we will be able to modulate this inflammatory niche and translate this into a meaningful hematological impact for our patient. And even, of course, we hope to translate it also in a kind of disease modulation. Ultimately, the success of inflammatory therapies in MDS will depend on identifying the right patients. So, the current prognostic scoring systems already incorporate parameters such as blast count, cytopenia, cytogenetics, molecular profiling, but the immunological features are still largely missing. So this is now beginning to change. There are initiatives such as the European Hematology Association Working Group, the EHA Working Group I for MDS. It's Immunology for MDS, which is led by Sharam Kordasti, which aims to better define the immune landscape of MDS patients and integrate these informations, these immune signatures into future classification and also treatment strategies. So with regard to S100A9 specifically, I think it's more complex than we think. So it's, of course, biologically highly relevant, but probably not straightforward, usable as a simple plasma-based biomarker. So in our own MDS cohort, circulating plasma S100 levels were not significantly different from healthy controls. Also, the S100A9 mRNA expression in the bone marrow mononuclear cells was highly elevated in MDS patients. So one possible explanation might be that S100A9 acts mainly locally in the bone marrow and is therefore not so obvious in the circulation, in the plasma levels detectable. Yes, so most likely we will need a broader biomarker strategy integrating inflammatory cytokines, bone marrow niche markers, signatures of innate immune activation to identify the patients in whom the S100A9-driven inflammatory axis is truly active and therapeutically targetable. Yes.
Okay. Or you could do retrospective analysis to find out those who did respond and then find out what those biomarkers were in those phase clinical trials. I did forget to ask, actually, in the HD13 mouse model, it does progress eventually to AML, I understand. Did you follow the mice long enough to see if TASC actually increased survival and diminished the latency of AML in any respects?
Well, so we focused on these 12 weeks treatment of mice, but we had a small group of mice which we continued to follow up for a longer time, and also before we had a very few animals which proceed to develop signs of leukemia, which we had to stop, of course, then for our experiment. But obviously, tasquinimod could not, at least in these very low number of animals, inhibit or
Prevent,
block
I guess, totally.
the progression to leukemia.
Okay.
But sorry,
okay.
but you have to understand that this mice model is a kind of programmed model. They will inevitably go to
AML.
MDS and AML.
Yeah.
So
yeah.
the question is whether this is really the best model to prove this hypothesis. Yes. So we are
I
looking
agree.
for
I
the
agree.
human study.
Okay. Yeah, I know. And I think, we shouldn't always conflate what we see in the mouse with what we see in the humans in terms
Yes.
of, you already mentioned quality of life at the beginning in terms of infection, transfusions. I'm hoping you're not doing bone strength through a machine press on the humans in your phase 1 clinical trials, but I'm assuming you're not going to be doing that. But, elderly people have falls risks, I know. So anything that can improve quality of life, I think, is excellent for the therapy. And again, Manja, you mentioned the pleiotropic effects of tasquinimod. In this case, we often want to have a very targeted agent, but perhaps the pleiotropic effects is beneficial for MDS patients because it hits at so many places at the same time. So I think that's very, very promising. I have one final question, which is I like to ask all the people that come onto the podcast. And that is, of course, that when you write the paper, this doesn't often follow the order of the events or the experiments or your thought processes of how you develop the project. And so I'm wondering, were there any challenges during the project or any really surprising points where you had an aha moment, which was quite interesting and unexpected during your writing and journey of this project?
Well, so the first challenge was to integrate the different biological systems, so hematopoiesis, trauma biology, inflammation, and the bone biology into one current story. And what you also have to have in mind when working with MSCs is their abundance in the bone marrow aspirate. So this is really, really low, and you have to expand those cells. If you expand in vitro MSCs, they might change not only their phenotype, but also other characteristics. And the major problem for us was that they lose the inflammatory signature in vitro during ex vivo expansion. So because the signal, for example, given by S100A9 turning on this inflammatory cascade within the MSCs is not furthermore in our system because we don't have the cells, neutrophils, macrophages, which secrete in vivo the S100A9. So that mean that we had to prime the MSCs in culture to again have this inflammatory phenotype and characteristics. So we used a recombinant protein to add into the culture system and to activate the TLR4 downstream signaling, resulting in NF-kappa B activation and the downstream activation of pro-inflammatory cytokines such as IL-1 beta, for example. And we could establish these in vitro model with the MSC culture, but of course we had to titrate the recombinant protein concentration and had to see that we obtain the right readouts to establish the system.
Yep.
So there were some technical challenges, and like in many translational studies, the work evolved iteratively, and one result generated the next question, and the final narrative became much clearer only in the retrospective observation. And in many ways, the project reinforced how important it is to study MDS not only as a malignant disease of hematopoietic cells, but as a disease of the entire bone marrow, because we, of course, performed all the experiments in which we show the impact of the MSCs also with hematopoietic cells alone. And we are really surprised how significant were the effects obtained by tasquinimod treatment when we had the MSCs in co-culture in comparison to HSC cultures alone. So that was a bit challenging, but also really exciting to develop the experiments and these preclinical study. I find that MSCs are one of the most impressive cell type in the bone marrow because they are so heterogeneous and so dynamic. That makes them a really fascinating cell type. They have really important functions in the bone marrow.
Yeah, a previously probably underappreciated part of the MDS puzzle,
for sure,
so to speak. And Katja, for you, what was something exciting as a physician for you, like in terms of the future? What's going to excite you for the next steps?
Yes, so when we look at the molecular heterogeneity of MDS, you know that there's not just one mutation. We have so many broad spectrum of different mutations and disease-driving mechanism. And we hope with this approach, targeting this inflammatory microenvironment, this could be an option for a broader group of patients, not only for these IDH1 mutated and so on. This is what drives us to go further. Of course, there were many disappointing moments also, as always. And of course, we also observed the other inflammatory drugs that has been studied in MDS, canakinumab as an interleukin-1 receptor inhibitor, IRAK4 inhibitors, which looked highly promising biologically, but ultimately failed to demonstrate some meaningful clinical benefit. But our hope is that by targeting the inflammatory cascade at a very, very early beginning, we may achieve better results for our patients. And of course, we also need this research in identifying which patients will ultimately benefit from these immunological treatments.
Beautiful. Well, thank you, Katja. Thank you, Manja, for joining us on the podcast today. It's a wonderful paper. I really like how it was a very strong collaborative effort as well between physicians, scientists, and the wonderful Bone Lab, obviously, which is close by, which is very impressive. I would like to encourage our readers to search out your paper at Hemisphere. It's a wonderful read, and I look forward to reading more about perhaps the phase 1-2 clinical trials in Hemisphere in a couple of years' time, perhaps, and we can get you back on the podcast and you can give us an update on what worked and hopefully what, I guess, a double negative, what didn't work. So it's a positive clinical trial. Thank you both for joining us today.
Thank you very much.
Thank you for giving us the opportunity. See you.
It's great.
Thank you for listening to this episode of the Hemisphere podcast. All of Hemisphere's content is open access and can be found at www.hemispherejournal.com. We hope you will join us for future podcast episodes.